Hello, welcome to my poster, Drug Repurposing, Identification, and Prioritization for Polycystic Kidney Disease by Gene Signature Reversion. My name is Elizabeth Wilk. I'm in the Lassane lab at the University of Alabama at Birmingham. So a little bit of background for ADPKD. It's one of the most prevalent monogenic human diseases. Its current only FDA-approved treatment is tolvaptan, which causes severe side effects including liver toxicity. So drug repurposing is a great option for PKD patients because we could reduce the time and cost significantly by looking at already FDA-approved drugs and just using them for this new indication. So the gene expression profiles from ADPKD patients and the preclinical models have very altered transcriptic signatures, which means that we can then take those and compare against FDA-approved drug signatures to find ones that may be the most inversely related so that they can be used to perhaps reverse the cellular phenotypes. So I have a little bit of information about the kind of main hallmarks of PKD and some of the driving mechanisms for those phenotypes. And then when we look at those disease signatures, we can find inversely related drug perturbation signatures. And from there, those drugs may very well work on those molecular mechanisms as mentioned in the first. So the methods for this, I used publicly available RNA-seq mouse PKD data and processed with NF-core RNA-seq pipeline. I used DE-seq2 for differential expression analysis. And then for functional enrichment analysis, I used cluster profile and gprofiler. I used signature search for signature reversion. And then to find FDA-approved drugs and mechanisms of action, I further annotated the drug data with Drug Repurposing Hub and OnSides data. So some of the results that I found were that for the upregulated and downregulated differentially expressed genes from this data, a lot of the pathways mapped to different immune pathways, as well as many for cell cycle, and then a few metabolism as well. And then further, I looked at the drug candidates for these profiles and looking at their gene targets, doing enrichment for those, also found a lot of pathways that are very relevant for PKD. So then again, looking at the top 30 drugs that are the most opposite to the PKD disease signature, and then looking at which drugs again are already FDA approved so that we could get them to patients sooner. These right here are colored in the mechanisms of action by their weighted connectivity score, which is like a type of enrichment score, again just looking at how inversely related those signatures are. They are starred by purple ones if their mechanism of action is very strongly associated with PKD. For example, the estrogen agonist. Estrogen has been seen to be protective for PKD, but hasn't actually been looked at for a treatment. And then the red-starred mechanisms of action are the ones that actually have been explored for possible treatments for PKD. I then have a little diagram for how one hypertension drug may be able to, while originally indicated for the vasodilation, may be used in PKD for reducing inflammation or preventing cell proliferation. So most of the developed drugs for PKD that are being worked on are Vaptans, which again have continued to show severe liver toxicity. So that there is a total of 82 already FDA approved drugs that came up with these with the signature reversion method. And then some of these mechanisms of action have been found for in the literature for PKD, as far as in one large drug screen. Topoisomerase inhibitors were found to reduce cyst growth, which were one of my top mechanisms of action here, and then tubulin inhibitors as well, without even affecting cell viability as well. So dopamine receptor antagonists are kind of a surprising result, but they have also been explored actually for HDAC5 nuclear export, and then for PKD as well. And then for phosphodiesterase inhibitors, specifically vinpocetine, has been researched for acute kidney injury in mice and rats, and it was found to reduce drug-induced oxidative stress, morphological changes, apoptosis, and cytokine production, and NFK beta activation. And I thank you to my lab and collaborators for all their help and input. Thank you.